Kentucky researchers announced that there's an updated Salmonella vaccine for horses. So what do we actually know? If you own horses, breed horses, race horses, or simply care about equine health, a recent announcement out of Kentucky may have caught your attention. Hagyard Equine Medical Institute, the University of Kentucky's Gluck Equine Research Center, and the Kentucky Thoroughbred Association have announced an updated version of an equine Salmonella vaccine. The announcement has been welcomed by many veterinarians because it addresses an emerging bacterial strain that researchers say has been appearing more frequently in horses in recent years. But while the announcement sounds promising, there is also an important caveat. At this point, very little detailed scientific evidence has been released publicly regarding how well the updated vaccine works. Today, we're going to break down exactly what was announced, why researchers felt an update was necessary, what makes this new Salmonella strain concerning, and why horse owners should remain cautiously optimistic until more data becomes available. So let's get started. To understand why this announcement matters, we first need to look at Salmonella. Many horse owners associate Salmonella primarily with food poisoning in people, but the bacteria can also cause serious disease in horses. Equine salmonellosis can range from mild diarrhea to severe life-threatening illness. In some cases, affected horses develop fever, dehydration, endotoxemia, and severe gastrointestinal damage. Hospitalized horses are particularly vulnerable, and outbreaks can spread rapidly through veterinary facilities, breeding farms, and training centers. What makes Salmonella especially challenging is that some horses can carry and shed the bacteria without showing obvious signs of illness. That means outbreaks can sometimes begin before anyone realizes there's a problem. For decades, veterinarians have worked to reduce the impact of Salmonella through biosecurity measures, sanitation protocols, isolation procedures, and vaccination strategies. In fact, this isn't the first time Kentucky researchers have tackled this issue. According to the announcement, roughly 15 years ago, researchers at the Gluck Equine Research Center and veterinarians at Hagyard collaborated to develop Salmonella and Clostridium vaccines in response to disease concerns within Kentucky's thoroughbred population. Those vaccines were considered an important advancement for equine health at the time. Now, that same partnership has returned to address what they believe is a new threat. According to Hagyard veterinarian Dr. Nathan Slovis, veterinarians have observed an increase in horses affected by Salmonella over the last several years, prompting efforts to improve the existing vaccine. The key development came through genetic analysis. Researchers at the Gluck Center used whole-genome sequencing to study Salmonella samples collected from horses. One particular strain caught their attention, Salmonella Saintpaul. Now, if you're familiar with bacterial genetics, here's why this is significant. Whole genome sequencing allows scientists to examine the complete genetic blueprint of an organism. It's essentially the microbial equivalent of reading every page of a book instead of just reading the chapter titles. By doing this, researchers discovered that many of the Salmonella Saintpaul samples were remarkably similar to one another genetically. In other words, they appeared to come from closely related bacterial populations. Even more concerning was what they found next. The Kentucky strains reportedly resembled Salmonella strains associated with outbreaks in Texas and Georgia during the 2023 season. Researchers also identified antibiotic-resistant genes within these bacteria. That phrase, antibiotic resistance, is enough to get the attention of any veterinarian. Antibiotic resistant occurs when bacteria evolve mechanisms that allow them to survive treatments that would normally kill them. This doesn't necessarily mean every infected horse becomes untreatable. However, it can limit treatment options and make disease control more difficult. For an industry that depends heavily on effective veterinary care, the emergence of resistant bacterial strains is something that deserves serious attention. The researchers described the pattern they observed as consistent with clonal expansion. Essentially, a successful bacterial lineage appears to have spread and multiplied while retaining most of its genetic characteristics. Once research identified this strain, attention shifted towards updating the vaccine. According to the announcement, 2 new Salmonella subspecies have now been added to the vaccine formulation. These additions were specifically selected to improve protection against circulating Salmonella infections. And this is where the story becomes both encouraging and somewhat frustrating. The encouraging part is that modern genomic technology appears to have been used exactly as many scientists hope it would be. Researchers identified an emerging pathogen. They characterized its genetics. They recognized potential antibiotic-resistant concerns, and they used that information to guide vaccine development. That's a powerful example of precision disease surveillance. In theory, it represents the future of veterinary medicine. Instead of waiting years to react to outbreaks, researchers can rapidly identify threats and develop targeted interventions. Dr. Yasura Hemley of the Gluck Equine Research Center described the project as an example of translating genomic data into practical protection for horses and the thoroughbred industry. Those are encouraging words, but here's the challenge. The public announcement contains very few details about the actual evidence supporting the updated vaccine. For example, what level of protection does the vaccine provide? How many horses were involved in efficacy testing? Were challenge studies conducted? What were the antibody responses? How long does immunity last? Does the vaccine specifically reduce shedding of Salmonella Saintpaul? Does it prevent infection entirely? At this point, those questions remain largely unanswered in publicly available materials. The announcement states that Hagyard veterinarians helped assess efficacy, but detailed results have not been yet released. That doesn't mean the vaccine isn't ineffective. Far from it. Many veterinary innovations begin with promising field data before formal peer-reviewed publication. However, from a scientific perspective, evidence matters. Veterinarians and horse owners ultimately need more than a press release. They need data, peer-reviewed studies, efficacy trials, safety assessments, and real-world outcome measurements will help determine just how valuable this updated vaccine truly is. This distinction is important because vaccines can vary considerably in performance. Some vaccines prevent infection. Others reduce disease severity. Others primarily reduce bacterial shedding and transmission. Each outcome can still be beneficial, but understanding exactly what a vaccine accomplishes is critical for making informed decisions. For breeding farms, racing operations, sales complex, and veterinary hospitals, those details can significantly influence disease control strategies. The announcement also highlights a broader trend within equine medicine. Historically, much of the infectious disease management focused on responding after outbreaks occurred. Today, advances in molecular biology are allowing researchers to identify emerging threats much earlier. Genomic surveillance has become increasingly important, not only for human medicine, but also for animal health. Whether we're talking about equine influenza, herpes virus, strangles, or bacterial pathogens like Salmonella, the ability to monitor genetic change in infectious organisms offers opportunities to stay ahead of disease rather than reacting to it. That may ultimately be one of the most important aspects of the story. The vaccine itself is important, but the process behind it, its development, may be equally significant. Researchers identified a changing pathogen landscape, applied advanced genomic tools, and modified a vaccine accordingly. That's exactly the kind of adaptive disease management many experts believe will be necessary as pathogens continue to evolve. Still, caution remains warranted. Until more detailed scientific information becomes available, it's difficult to determine exactly how much additional protection this updated vaccine provides. For now, horse owners should view this development as promising but preliminary. It's encouraging to see collaboration between private practitioners, academic researchers, and industry organizations. It's encouraging to see genomic surveillance leading directly to practical interventions. And it's certainly encouraging to see attention focused on a disease that can cause serious consequences for horses and equine facilities. But as always in science, the strongest conclusions come from evidence. The next step will be publication of efficacy data, safety information, and field results that allow veterinarians and horse owners to evaluate the vaccine's performance objectively. Until then, this announcement represents an interesting and potentially important advancement in equine disease prevention, but one that still raises as many questions as it answers. And that may be the most honest takeaway from this story. A potentially valuable new tool has arrived. Now the industry waits to see the data. If you found this helpful, please feel free to join and subscribe. Thank you so much for joining us. And remember, I am not AI. If you listen closely, you can hear me breathing.